of you are doing well. I am Honey Rachel. I teach Biotechnology in K.B.N college. In this session, let us try to learn something interesting. Before I start, I want you to observe these pictures. This one, rice, a cow, cotton, and chicken. They look normal, right? All of these. This looks quite different. But these, let me tell you, these are not normal. These are quite different from normal chicken. This chicken you see is a disease resistant chicken which is resistant to lot of number of fungal, bacterial and viral diseases. And this cotton is not a normal cotton at all. This cotton is pathogen resistant cotton which fights all sorts of pests and gives wonderful yield, I must say. And this cow, it has double the muscle of normal cow. And this rice is golden rice. It has vitamin A very rich in it. All these are quite unique, right? Do you know how all these are made? Yes, you guessed it right. All these are genetically engineered organisms. They have special genes in them. Now, the question is how these genetically engineered organisms are made. Shall we try to understand in this session what is genetic engineering, how genetically modified organisms like chicken, cotton, and many more are made? Yes, let us understand what genetic engineering is first. Genetic engineering is nothing but inserting a desired or special gene into a specific crop so that we get in that crop the special characters. Like in golden rice, we have inserted a gene which helps in synthesizing vitamin A in the rice itself so that whenever we consume this golden rice, we get rich vitamin A. And all other GMOs have special characters because of a special genes which was once inserted into the crop or animal. We can derive genetically modified organisms of different kinds. We can get genetically modified plants, animals as we have seen earlier, and not only plants and animals, we can also get genetically modified microbes like genetically modified bacteria. The insulin that we take every day is actually produced from a genetically modified microbe. Let us understand more how the genetic engineering works, the process of genetic engineering. Shall we? Yes. For genetic engineering, first we need to have the desired gene, a special gene, like in golden rice, the gene that can synthesize vitamin A. So we will take the cell which has that special gene or desired gene, and then we will lysis the cell. We will break down the cell, collect all the genome present in the cell, like you see here. This red color thing is the genome, the whole DNA of that particular cell. We will extract that using different cell lysis methods, physical, chemical methods of cell lysis, enzymatic as well. As we get the whole genome, we do not want whole genome. We want only one specific gene. For example, we want only vitamin A synthesizing gene. So what we'll do is we will cut the DNA into small fragments. How do we do that? We cut the DNA into small fragments using restriction enzymes. So remember restriction enzymes. These are wonderful molecular scissors. These are present in bacteria. So with the help of restriction enzymes, we cut the DNA, yes, this DNA, isolated one, into many small fragments. After we get many small fragments, we will separate them because out of all of that cluster of genes, we want only one gene. So first we will isolate and then we will separate. As we separate, we use the technique electrophoresis, a gross gel electrophoresis, which is ideal for separating DNA molecules depending upon their size. Shorter length DNA will isolate first, will come first in the race of electrophoresis, and the longer fragment comes last. Like that, as we separate the DNA fragments depending on their length using electrophoresis, we get number of bands. Out of that, we will select the desired DNA depending upon the length of base pairs. And now voila, we have the desired DNA with desired length. As we have only one copy of desired DNA, what we'll do is we will multiply that into several copies. Which technique do we use for this is DNA amplification, PCR. After PCR or after DNA amplification, we get number of copies of DNA of desired origin. So with those number of copies, we can go further for the genetic engineering process. Now the first step of genetic engineering is complete, that is preparation of desired gene. Now we have number of copies of one particular desired gene. These are the copies. Now we can go for the second step, that is isolation of DNA vector. What is this DNA vector? It's a circular DNA strand, simple one with some genetic markers with a replication site, origin of replication. Usually vectors are plasmids. They are present in bacteria. And why do we need this vector? Is because as we want to insert the gene of interest, the desired gene, into the host cell, we need a vehicle to carry our desired gene. Yes, we of course need a vehicle. Why? Because if we insert the DNA directly into the cell might reject the DNA because it is foreign. It doesn't belong to the host cell. That's why we need a carrier which imitates like ohm of host cell and which transports as a vehicle, carefully, precisely delivers the desired gene into our host cell. And this circular round thing is plasmid, a vector, an example of DNA vector. Yes, usually we find these vectors, especially plasmids, in bacteria. In bacteria, they exist as extra chromosomal DNA. So what we need to do? We need to isolate that vector and attach our desired gene with that vector. For that, in the second step of our genetic engineering, what we do is we do cell lysis. We break the cell of bacteria, we isolate the vector, that is plasmid, and then we will cut it at restriction site. The same enzyme which we use to cut this DNA, desired DNA, we'll use the same enzyme to cut our vector, plasmid, and then we get the cleavage. In that cleavage, we would want to insert this gene and make it a recombinant DNA. So with this cleavage and with this fragment, who will help us attach these two is a wonderful enzyme called DNA ligase. Yes, DNA ligase acts as a molecular glue and sticks both the desired gene as well as our vector. And then as we use DNA ligase and combine the two, the fragment as well as the vector, we get a recombinant DNA. We call it recombinant because it has an foreign gene in it and it is recombinant. Yes, we finally have the desired gene inserted into the vector. Now we just have to insert that vector. Sir, insert that recombinant DNA into the host cell. The host cell can vary. It can be a plant, it can be an animal cell, it can be a bacterial cell as well. In this picture, we were just focusing more on bacterial recombination. So, as a host cell, so we took bacterial cells, and in the fourth step, we are inserting the recombinant gene. How do we do that? We use several gene transfer methods, physical methods like microinjection, particle bombardment, or biological methods like transfection, transformation, chemical methods like liposome mediated gene transfer. Through all these gene transfer methods, we will insert the recombinant DNA into our host cell. As the host cell accepts the recombinant DNA, do you know what it becomes? It becomes a recombinant cell. Yes, a cell which contains recombinant DNA is called recombinant cell. By that, we complete the fourth step, gene transfer. But how do we know whether all cells accepted the gene or not? Sometimes few cells doesn't accept the gene. Sometimes few cells accept the gene. So how do we separate the cells which accepted and which did not accept? We do a screening test. That is fifth step of the process, the selection of recombinant cells. So for that purpose, what do? We take the cells, recombinant cells, and grow them in a special medium. In a petri dish. And on that, we get two types of colonies, blue and white. Yes, if you see it here, yes, we have both blue circles and yellow circles. The blue colonies are non-recombinant, the cells that do not have any desired DNA. The white colonies are those cells which have our recombinant DNA. So here in this step, we select only white colonies. We select only those cells which have recombinant DNA, and then finally we use all those cells to produce a desired protein or to create a plant or animal with desired gene, transgenics. Yes. Do you know all these animals and plants, they are called transgenics. They have special gene in them. Shall we go further? Yes. In this process, there are two key elements. The restriction enzymes, which helps in digesting the DNA or cutting the DNA at specific sites, leaving blunt or sticky ends. And another element, the most important one, is cloning vector. Yes, we have restriction enzymes like EcoR1, BAMH1. We also have cloning vectors like PBR322. Let us study more about these tools of genetic engineering in the next session. I hope you enjoyed this session. Thank you.